Hello everyone, welcome to the next lecture in our Physiology video lecture series. This particular lecture is titled The Nervous System, General Principles and Sensory Physiology. In this particular lecture, we will briefly go through the nervous system, the general principle by which it functions, the sensory and integrative and motor functions of the nervous system as well. So let us get started. First, let us look at the general design of the nervous system. As we know, the basic functional unit of the nervous system is the neuron. And the central nervous system contains more than 100 billion neurons. The figure that is shown right here shows a typical neuron of a type found in the brain motor cortex. Incoming signals enter this neuron through synapses located mostly on the neuronal dendrites, but also present on the cell body. So dendrites are the small extension from the cell body, and the cell body itself is the part of the neuron that houses the nucleus. It does not normally have any extension on itself. And as we have already discussed in the first lecture, or first or second lecture of our whole video lecture series, dendrites are the places where incoming signals are received by a neuron, and axon is the largest protrusion or extension of the cell body of the neuron, which is shown right here. And this axon is the one which sends signal out of the neuron. Thus, the incoming end of nervous signals is the dendrite, and the outgoing end is the axon. For different types of neurons, there may be only a few hundred or as many as 200,000 such synaptic connections from input fibers. Conversely, the output signal travels by way of a single axon leaving the neuron. Then, this axon has many separate branches to other parts of the nervous system or peripheral body. A special feature of most synapses is that the signal normally passes only in the forward direction, that is, from the axon of a preceding neuron to dendrites on the cell membrane of subsequent neuron. That is, in the diagram, the direction will be from upwards to downwards. This forces the signals to travel in required directions for performing specific nervous functions. Next, we look into the sensory parts of the nervous system, which are the sensory receptors. Most activities of the nervous system are initiated by the sensory experience exciting sensory receptors, whether visual receptors in the eyes, auditory receptors in the ears, tactile receptors on the surface of the body, or other kind of receptors. This sensory experience can either cause immediate reaction from the brain, or memory of the experience can be stored in the brain for minutes, weeks, or years and determine bodily reactions at some future date. The figure that is shown right here shows the somatic portion of the sensory nervous system, which transmits sensory information from receptors of the entire body surface and from some deep structures. So, somatic portion means the sensory portion or the sense of touch. So, the receptors that are shown here, which include the Meissner's corpuscles, that some of which we will discuss in the later part of the slides, respond to touch, and the signal is then transmitted to the somatosensory cortex that is present in our brain. This information enters the central nervous system through peripheral nerves and is conducted immediately to multiple sensory areas. In first, the spinal cord at all levels, the reticular substance of the medulla pons and mesencephalon of the brain, the cerebellum, the thalamus, and areas of the cerebral cortex. Now that was the affected part of the central nervous system. That is the part that acknowledges the presence of an external stimulus. But a second part, which is the effective part, that is what the response of the body toward the stimulus will be, is determined by a separate portion of the central nervous system, which is the motor part of the nervous system. The most important eventual role of the nervous system is to control the various bodily activities. This is achieved by controlling first the contraction of appropriate skeletal muscles throughout the body, two, the contraction of smooth muscles in the internal organs, three, the secretion of active chemical substances by both exocrine and endocrine glands in many parts of the body. These activities are collectively called motor functions of the nervous system, and the muscles and glands are called effectors because they are the actual anatomical structures that perform the functions dictated by the nerve signals. That is, these particular parts of the body are the ones that respond to the stimulus that was received by the nervous system or the sensory part of the nervous system, and now the motor part will actively respond to it. The figure right here shows a skeletal motor nerve axis of the nervous system for controlling skeletal muscle contraction. Operating parallel to this axis is another system called the autonomic nervous system for controlling smooth muscles, glands, and other internal bodily systems. The skeletal muscles can be controlled from many levels of the central nervous system, including the spinal cord, the reticular substance of the medulla, pons, and mesencephalon, the basal ganglia, the cerebellum, and the motor cortex. So here we see that more parts of the brain can effectively control the motor part of the nervous system, and the sensory information is processed in only in a little bit smaller parts of the brain. Each of these areas play its own specific role. The lower regions concern primarily with automatic, instantaneous muscle responses to sensory stimuli, and the higher regions with the deliberate, complex muscle movements controlled by the thought processes of the brain. Next, we move on to one of the most important functions of the nervous system, that is processing of information and the integrative function of the nervous system. More than 99% of all sensory information is discarded by the brain as irrelevant and unimportant. For instance, one is ordinarily unaware of the parts of the body that are in contact with clothing, as well as the seat pressure when sitting. Likewise, attention is drawn only to an occasional object in one's field of vision, and even the perpetual noise of our surroundings is usually relegated to the subconscious. That is, the brain does not actively process every bit of information or stimuli that it is presented to. But when it's important sensory information excites the mind, it is immediately channeled into proper integrative and motor regions of the brain to cause desired responses. This channeling and processing of the information is called the integrative function of the nervous system. Next, we look into the role of synapses in this processing of information. The synapse is the junction point from one neuron to the next. Synapses determine the direction that the nervous signals will spread through the nervous system. Some synapses transmit signals from one neuron to the next with ease, whereas others transmit signals only with difficulty. Also, facilitatory and inhibitory signals from other areas of the nervous system can control synaptic transmission, sometimes opening the synapses for transmission and at other times closing them. Thus, synapses perform a selective action, often blocking weak signals while allowing strong signals to pass, but at other times selecting and amplifying certain weak signals and often channeling these signals in many directions rather than only in one direction. Thus, the synapses can play a very important role in selecting and filtering the particular signals that will be passed through it, and thus can help in processing of specific important information while ignoring the overall stimulus that is received by the body. Next, we look into the storage of information in the form of memory. Only a small fraction of even the most important sensory information usually causes immediate motor response. But much of the information is stored for future control of motor activities and for use in the thinking process. Most storage occurs in the cerebral cortex, but even the basal regions of the brain, the spinal cord, and can store small amounts of information. The storage of information is the process we call memory, and this too is a function of synapses. That is, each time certain types of sensory signals pass through sequences of synapses, these synapses become more capable of transmitting the same type of signal the next time, a process called facilitation. And thus, as more and more stimulation of a particular synapse is done, the signal that is now transmitted through that synapse is facilitated, or it can move much easily through it the next time. And thus, in this form, memory is developed in the nervous system. That is, the next time a particular stimulus is received, it will be facilitated to move through a particular pathway, and thus the body can effectively respond to the stimulus in a pre-described manner. Next, we move into major levels of the central nervous system. The human nervous system has inherited special functional capabilities from each stage of human evolutionary development. From this heritage, three major levels of central nervous system have specific functional characteristics. These levels include the spinal cord, the lower brain or subcortical brain, and the higher brain or cortical level. First, we'll look into the spinal cord. Even after the spinal cord has been cut in the high neck region, many highly organized spinal cord functions still occur. For instance, neuronal circuits in the cord can cause walking movements, reflexes that withdraw portions of the body from painful objects, reflexes that stiffen the legs to support the body against gravity, and reflexes that control local blood vessels, gastrointestinal movements, or urinary excretion. The upper levels of the nervous system often operate not by sending signals directly to the periphery of the body, but by sending signals to the control centers of the cord, simply commanding the spinal cord centers to perform their functions. And thus, the function is essentially performed at the spinal cord level and not at higher brain levels. Next, we look into the lower brain or subcortical level. Many, if not most, of what we call subconscious activities of the body are controlled in the lower areas of the brain. In the medulla, pons, mesencephalon, hypothalamus, thalamus, cerebellum, and basal ganglia. For instance, subconscious control of arterial pressure and respiration is achieved mainly in the medulla and pons. Feeding reflexes such as salivation and licking of lips in response to taste of food are controlled by areas in the medulla, pons, mesencephalon, amygdala, and hypothalamus. And many emotional patterns such as anger, excitement, sexual response, reaction to pain, reaction to pleasure can still occur after destruction of much of the cerebral cortex. Next, we look into the higher brain or cortical level of the nervous system. Since many of the nervous system functions occur at the cord and lower brain levels, one may be curious what is left for the cerebral cortex to do. The answer to this complex question, the answer to this is complex, but it begins with the fact that the cerebral cortex is an extremely large or portion of the nervous system. Without the central cerebral cortex, the functions of the lower brain centers are often imprecise. The vast storehouse of cortical information usually converts these functions to determinative and precise operations. So, one may imagine this higher brain or cortical level of the central nervous system as the part of the nervous system that controls the lower brain or supervises the lower brain and the spinal cord levels. Without the presence of the cortical level of the nervous system, many of the functions that could have been performed by the spinal cord level or the lower brain level do not sync and are imprecise in nature. And it is also a storehouse of memory, which helps it in performance of its various tasks. Next, we will look at the central nervous system synapses. Information is transmitted in the central nervous system mainly in the form of nerve impulses, that is, through a succession of neurons one after another. Nerve impulses are obviously the action potentials that we have already discussed way before in probably our third or fourth lecture. And that part is something that will play a major role in subsequent slides. So if we do not recall it very well, then we can obviously visit the previous lecture on this particular channel. However, in addition, each impulse may be blocked in its transmission from one neuron to the next, may be changed from single impulse to repetitive impulses, or may be integrated with impulses from other neurons to cause highly intricate patterns of impulses in successive neurons. All these functions can be classified as synaptic functions of neurons. Now we'll look into the types of synapses that we have been talking about for the last few minutes. The types of synapses are divided into two major halves. The first is the chemical synapse and the second is the electrical synapse. Almost all synapses used for signal transmission in the central nervous system of the human being are chemical synapses. In these, the first neuron secretes at the nerve ending synapse a chemical substance called a neurotransmitter. And this transmitter in turn acts on receptor proteins in the membrane of the next neuron to excite the neuron, inhibit it, or modify its sensitivity in some other way. And thus, in a chemical neurons, chemical substances known as neurotransmitters such as acetylcholine are involved majorly in transmission of the action potential or nerve impulses. But in the case of electrical synapses, however, are characterized by direct open fluid channels that conduct electricity from one cell to the next. So in case of chemical junctions or chemical synapses, there is open space between two neurons, and the presynaptic neuron secretes a particular neurotransmitter which signals the postsynaptic neurons to exhibit the action potential, and this is how the nerve impulse in the form of electricity is transferred from the presynaptic neuron to the postsynaptic neuron. But in the case of electrical synapses, the presynaptic neuron and the postsynaptic neuron are connected by special junctions called gap junctions, which are essentially pores between the present in the membranes of the two neurons, the presynaptic and the postsynaptic ones, functioning effectively as tunnels or pores through which the cytoplasm of the presynaptic and the postsynaptic neurons are conjoined or connected. Thus, when action potential occurs in the presynaptic neuron, there is no need of any neurotransmitter to be released, as action potential can easily travel through the adjoining membrane and the cytoplasm of the presynaptic neuron to the postsynaptic neuron. And this is how electrical synapses function. Most of these most of these direct fluid channel consist of small protein tubular structures called gap junctions that allow free movement of ions from the interior of one cell to the interior of the next. And thus, with the free movement of ions, the action potential is also transferred from one neuron to the next, and this is how nerve impulses are transmitted in the case of electrical synapses. Next, we look at important characteristic of chemical synapses, which is one-way conduction. Chemical synapses have one exceedingly important characteristic that make them highly desirable for transmitting most nervous system signals. That is, they always transmit the signals in one direction. That is from a neuron that secretes the transmitter substance called presynaptic neuron to the neuron on which the transmitter acts, called the postsynaptic neuron. As we have already discussed in the first slide, in case of chemical synapses, there is a specific direction in which the nerve impulse can travel, that is from the dendrite to the axon of the postsynaptic neuron. And thus, this allows for only specific one-way conduction of nerve impulses, and this particular characteristics is highly desirable in nervous system. Next, we look into the physiologic anatomy of the synapse. The figure shows an anterior motor neuron in the anterior horn of the spinal cord. It is composed of three major parts: the soma, which is the main body of the neuron or the cell body of the neuron which houses the nucleus, a single axon which extends from the soma into the peripheral nervous system and leaves the spinal cord, and the dendrites, which are the great numbers of branching projections of the soma that extend as much as 1 millimeter into the surrounding area of the cord. As many as 10,000 to 200,000 minute synaptic knobs called presynaptic terminals lie on the surface of the dendrites and soma of the motor neurons. About 80 to 95% of them are present on the dendrites and only 5 to 20% on the soma. These presynaptic terminals are the ends of the nerve fibrils that originate from many other neurons. These presynaptic terminals are the locations from which the incoming nervous impulse is received by the postsynaptic neuron. And we will look into detail how this occurs in the next slide. Let us look at first at the presynaptic terminals. That is this neuron which comes into the picture from the above end is the presynaptic neuron, which releases the neurotransmitter, and this neuron that is shown here is the postsynaptic neuron, which the neurotransmitter acts. We are first looking at the presynaptic terminal of the synapse, that is a portion of the presynaptic neuron. The figure here illustrates the basic structure of a synapse, showing a single presynaptic terminal on the membrane surface of a postsynaptic neuron. The presynaptic terminal is separated from the postsynaptic neuronal soma by a synaptic cleft, which has a width of usually 200 to 300 angstrom. The terminal has two internal structures important to excitatory or inhibitory functions of the synapse, which are the transmitter vesicles and the mitochondria. The transmitter vesicles contain the transmitter substance that, when released into synaptic cleft, either excites or inhibits the postsynaptic neuron. The neurotransmitter will excite the neuronal membrane if the neuronal membrane contains excitatory receptors, and inhibit the membrane if it contains inhibitory receptors. The mitochondria provides adenosine triphosphate, that is ATP, which is the energy currency of the cell, which in turn supplies energy for synthesizing the new transmitter substance. And thus, these two, that is the transmitter vesicles and the mitochondria, go hand in hand in order for transmission of nervous signals. Next, we'll look into detail into the mechanism by which an action potential causes transmitter release from presynaptic terminals, which essentially depends on the role of calcium ions. The membrane of the presynaptic terminal is called the presynaptic membrane. It contains large numbers of voltage-gated calcium channels. When an action potential depolarizes the presynaptic membrane, these calcium channels open and allow large numbers of calcium ions to flow into the terminal. The quantity of transmitter substance that is then released from the terminal into the synaptic cleft is directly related to the number of calcium ions that enter. The precise mechanism by which the calcium ions cause this release is not yet known, but it is believed or speculated to be the following. When the calcium ions enter the presynaptic terminal, they bind with special protein molecules on the inside surface of the presynaptic membrane called release sites. This binding in turn causes the release sites to open through the membrane, allowing a few transmitter vesicles to release their transmitter into the cleft after each action potential. For those vesicles that store neurotransmitter acetylcholine, between 2000 to 10,000 molecules of acetylcholine are present in each vesicle, and there are enough vesicles in the presynaptic terminal to transmit from a few hundred to more than 10,000 action potentials. Next, we look into the action of transmitter substances on the postsynaptic neuron, which is essentially a function of receptor proteins that receive the neurotransmitter that is released by the presynaptic neuron. The membrane of the postsynaptic neuron contains a large number of receptor proteins, which are also shown in the picture. These red dots are the receptor proteins. The molecules of these receptors have two important components. The first one is a binding component that protrudes outward from the membrane into the synaptic cleft. Here it binds to the neurotransmitter coming from the presynaptic terminal. The second is an ionophore component that passes all the way through the postsynaptic membrane to the interior of the postsynaptic neuron. The ionophore, in turn, is one of two types. It can either be an ion channel that allows the passage of specified types of ions through the membrane, or a secondary messenger, activator that is not an ion channel but instead is a molecule that protrudes into the cell cytoplasm and activates one or more substances inside the postsynaptic neuron. These substances in turn serve as second messengers to increase or decrease specific cellular functions. Next, we will look into detail in what exactly are these ion channels and the second messenger system. The ion channels in the postsynaptic neuronal membrane are usually of two types. The first are cationic channels that most often allow sodium ions to pass through when open, but sometimes can also allow the passage of potassium and calcium ions as well. The second type of ion channels are the anion channels that allow mainly chloride ions to pass through, but also minute quantities of other anions. When cation channels are open and allow positively charged sodium ions to enter, the positive electrical charges of the sodium ions will in turn excite this neuron. Therefore, a transmitter substance that opens cation channels is called an excitatory transmitter or an excited transmitter. Conversely, opening anion channels allows negative electrical charges to enter, which inhibits the neuron. Therefore, transmitter substances that open these channels are called inhibitory transmitters. The reason why the entry of positive ions is excitatory or the negative ions is inhibitory is also discussed in our previous lectures, in which we have discussed membrane potential and how membrane potential depends on passage of specific ions through the membrane. I will recommend watching that video again in case we are having any problem understanding all this. Next, we look into the second messenger system, the postsynaptic neuron. Many functions of the nervous system, for instance, the process of memory, require prolonged changes in neurons for seconds, from seconds to months after the initial transmitter substances is formed. That is, there needs to be a memory of the particular neural of the particular nervous impulse stored in the cell. This cannot be done by normal ion channels or any other way, and is handled by the second messenger system. One of the most common second messenger systems uses a group of proteins called G proteins. G proteins consist of three components: an alpha component, that is the activator portion of the G protein, a beta and a gamma component that are attached to the alpha component and also to the inside of the cell membrane adjacent to the receptor protein. On activation by nerve impulse, the alpha portion of the G protein separates from the beta and gamma portion, which is shown right here in this schematic, and then is free to move within the cytoplasm of the cell. Inside the cytoplasm, the separated alpha component performs one or more of multiple functions, which depends on the specific characteristic of the neuron. These particular functions can be opening specific ion channels through postsynaptic cell membrane, activation of cyclic adenosine monophosphate or CAMP or cyclic guanosine monophosphate or CGMP in the neuronal cell, activation of one or more intracellular enzymes, or activation of gene transcription. That thus, after receiving the initial signal, that is the nerve impulse, the secondary messenger system will transmit or will carry on this particular signal from the and convert the particular nervous impulse into special responses by the neuronal cell. These responses depend on the particular nervous cell which we are talking about, and can include variety of responses, which can include opening of specific ion channels, to transmission of the secondary message via other molecules such as CAMP, CGMP, which can ultimately cause gene activation as well. Next, we look into the excitatory and inhibitory receptors that we are talking about, which are present in the postsynaptic membrane. Some synaptic receptors, when activated, cause excitation of the postsynaptic neuron, and others cause inhibition. The importance of having inhibitory as well as excitatory types of receptors is that this gives an additional dimension to nervous functions, allowing restraint of nervous action as well as excitation. So first, let us look at excitation or excitatory receptors which are present on the postsynaptic membrane. Excitation of a particular nervous cell can include opening of sodium channels to allow large numbers of positive electrical charges to flow into the interior of the postsynaptic cell. This raises the intracellular membrane potential in the positive direction up towards the threshold level for excitation. It is by far the most widely used means for causing excitation. Once the membrane potential has risen closer to the threshold membrane potential for an action potential, the stimulation of the nervous cell or the stimulation of the neuron required for it to actually exhibit action potential is far less, and thus it can be very easily excited than in normal conditions. Depressed conduction through chloride or potassium channels of both. Depressing or reducing the conduction through chloride and potassium channels decreases the diffusion of negatively charged chloride ions to the inside of the postsynaptic neuron, as well as decreasing the diffusion of positively charged potassium ions to the outside. In either instance, the effect is to make the internal membrane potential more positive than normal, which is excitatory in nature. Next point is the various changes in the internal metabolism of the postsynaptic neuron are also done by the excitatory signal. These various changes excite cell activity or can in some cases increase the number of excitatory membrane receptors or decrease the number of inhibitory membrane receptors. Next, we look into the inhibition of the postsynaptic neuron. This can happen via opening of chloride ion channels to the postsynaptic neuronal membrane. This allows rapid diffusion of negatively charged chloride ions from outside the postsynaptic neuron to the inside, thereby carrying negative charges inward and increasing the negativity inside, which is inhibitory in nature. It can also include increase in conductance of potassium ions out of the neuron. This allows positive ions to diffuse to the exterior, which causes increased negativity inside the neuron and thus increases the threshold of membrane potential required to be reached in order for an action potential to occur, and thus is inhibitory in nature. Finally, inhibition can be via activation of receptor enzymes that inhibit cellular metabolism functions that increase the number of inhibitory synaptic receptors or decrease the number of excitatory receptors. Next, we look into the effect of synaptic excitation on the postsynaptic membrane, that is excitatory postsynaptic potential. The figure A that is shown right here shows the resting neuron with an unexcited presynaptic terminal resting on its surface. The terminal is right here, which is left to right in the particular schematic. The resting membrane potential everywhere in the soma is minus 65 millivolts. Figure B shows a presynaptic terminal that has secreted an excitatory transmitter into the cleft between the terminal and the neuronal somal membrane. This particular presynaptic terminal is releasing excitatory neurotransmitter substance in between the presynaptic and the postsynaptic neuron inside the synaptic cleft. This transmitter acts on membrane excitatory receptors to increase the membrane's permeability to sodium ions. This rapid influx of positively charged sodium ions to the interior neutralizes part of the negativity of the resting membrane potential. As we know, the resting membrane potential of a neuron is always negative in nature. Thus, in the figure B, the resting membrane potential has increased in the positive direction from minus 65 to minus 45 millivolts. This positive increase in the voltage above the normal resting neuronal potential, which is around minus 90 millivolts, that is to a less negative value, is called excitatory postsynaptic potential or EPS. Next, we look into the generation of action potential in the initial segment of the axon leaving the neuron, which has a low threshold for excitation. When the EPSP rises high enough in the positive direction, there comes a point at which this initiates an action potential in the neuron. However, the action potential does not begin adjacent to the excitatory synapses. Instead, it begins in the initial segment of the axon, where the axon leaves the neuronal soma. The main reason for this point of origin of action potential is that the soma has relatively few voltage-gated sodium channels in its membrane, which in turn makes it difficult for the EPSP to open the required number of sodium channels to elicit an action potential. Conversely, the membrane of the initial segment has 7 times as great a concentration of voltage-gated sodium channels as does the soma, and therefore can generate an action potential with much greater ease than can the soma. The EPSP that will elicit an action potential in the axon initial segment is between plus 10 to plus 20 millivolts. This is in contrast to the plus 30 to plus 40 millivolts or more required on the soma. Once the action potential begins, it travels peripherally along the axon and usually also backward over to the soma. In some instances, it travels backward to as far as the dendrites, but not into all of them because they, like the neuronal soma, have very little or very few voltage-gated sodium channels and therefore cannot frequently generate action potentials at all. So we can see that the directionality of signal transduction in neuron is modified at the level of receptor proteins or a voltage gated or rather voltage gated channels that are present throughout the neuron. The voltage gated sodium channels that are required and are essential for creation of action potential are much more frequently observed on the axon part than the neuronal soma and dendrites. The number of voltage gated sodium channels on the axon is 7 times as great as that present in the neuronal soma. And thus, it is far easier to elicit action potential in the axon, giving rise partially to the directionality of the signal transduction rather than the action potential being generated in the neuronal soma on the dendrites. Next, we look into the electrical events during neuronal inhibition. First we look into the effect of inhibitory synapses on the postsynaptic membrane. That is the inhibitory postsynaptic potential. The inhibitory synapses open mainly chloride channels, allowing easy passage of chloride ions. Now, to understand how exactly the inhibitory synapses inhibit the postsynaptic neuron, we must recall what we learned about nurse potential of chloride ions, which is something we have gone through in the third or fourth lecture in our video lecture series. We have already calculated nurse potential for chloride ions to be about minus 70 millivolts. This potential is more negative than the minus 65 millivolts normally present inside the resting neuronal membrane. Therefore, opening chloride channels will allow negatively charged chloride ions to move from the extracellular fluid to the interior, which will make the interior membrane potential more negative than normal, approaching to minus 70 millivolt level. Opening potassium channels will allow positively charged potassium ions to move to the exterior, and this will also make the interior membrane potential more negative than usual. Thus, with chloride influx and potassium efflux increases the degree of intracellular negativity, which is a phenomenon known as hyperpolarization. Therefore, an increase in negativity between the normal resting membrane potential level is called an inhibitory postsynaptic potential or IPSP, as opposed to the excitatory postsynaptic potential, which was EPSP. Now we look into how exactly this causes a presynaptic inhibition. In addition to the postsynaptic inhibition, that is inhibition of neural transduction in the postsynaptic neuron, The presynaptic neuron can also be inhibited. In addition to the postsynaptic inhibition, another type of inhibition often occurs at presynaptic terminals even before the signal even reaches the synapse. This type of inhibition is called the presynaptic inhibition, occurs in the following way. So first we have to understand we are talking about inhibition of the presynaptic neuron, which was bringing the action potential and the neural impulse towards the postsynaptic neuron. The presynaptic inhibition is caused by release of an inhibitory substance onto the outside of the presynaptic nerve fibrils before their own endings terminate on the postsynaptic neuron. In most instances, the inhibitory transmitter substance is GABA, which is gamma aminobutyric acid. This has a specific effect of opening anion channels, allowing large numbers of chloride ions to diffuse into the terminal fiber. The negative charges of these ions inhibit the synaptic transmission because they cancel much of the excitatory effect of the positively charged sodium ions that also enters the terminal fibrils when an action potential arrives. And thus the mutual cancellation of the presynaptic inhibition and the action potential in the postsynaptic neuron cause the signal not to be transducted towards the postsynaptic neuron, and thus the neuronal impulse does not pass and is inhibited. Next, we look into how the simultaneous summation of the inhibitory and the excitatory postsynaptic potentials help in facilitation of neurons. Often, the summated postsynaptic potential is excitatory but has not risen high enough to reach the threshold for firing by the postsynaptic neuron. When this happens, the neuron is said to be facilitated. That is, its membrane potential is nearer the threshold for firing than normal, but not yet at the firing level. Consequently, another excitatory signal entering the neuron from some other source can then excite the neuron very easily. Diffuse signals in the nervous system often do facilitate a large number of neurons so that they can respond quickly and easily to the signals arriving from other sources. Let us now look into what is exactly facilitation of neurons. If an IPSP is tending to decrease the membrane potential to a more negative value, while an EPSP is tending to increase the potential at the same time, these two effects can either completely or partially nullify each other. Thus, if a neuron is being excited by an EPSP, an inhibitory signal from another source can often reduce the postsynaptic potential to less than the threshold value for excitation, thus turning off the activity of the neuron. So essentially, the whole mechanism of facilitation functions as if we can better explain via example. Say we have a parcel that we need to deliver to the 10th floor of a particular building. The excitatory postsynaptic potential, what the excitatory postsynaptic potential does is that it essentially helps the parcel reach the 5th or the 6th floor. What this does is that it does not deliver the parcel or the neuronal impulse to the postsynaptic neuron that is present on the 10th floor, but it helps it along. So that when the next push or the next excitatory postsynaptic, when the next EPSP arrives, it can then easily push the particular neuronal impulse to the 10th floor. that is to the postsynaptic neuron, and the postsynaptic neuron can then have an action potential. But this is inhibited by the IPSP, which instead, in this particular example, we can imagine is push the parcel down into the basement level. And thus, the next EPSP has to bring it up directly to the 10th floor plus the number of levels that it went below the ground floor. That is, it has to work additionally hard in order to excite the postsynaptic neuron. This is essentially how the whole facilitation of neurons. Next, we look into the sensory receptors and neuronal circuits for processing information. There are five basic types of sensory receptors, which include mechanoreceptors, which detect mechanical compression or stretching of receptors or of tissues adjacent to the receptors. These include a Pacinian corpuscle, Merkel's discs, or free nerve endings. Thermoreceptors, which detect changes in temperature, where some receptors can detect cold and other can detect warmth. These include cold receptors and warm receptors. Nociceptors, which are essentially pain receptors, which detect damage occurring to tissues, whether physical damage or chemical damage. These include free nerve endings. Electromagnetic receptors, which detect light on the retina, which obviously are the rods and the cones. And finally, the chemoreceptors, which detect the taste in our mouth, smell in our nose, the oxygen level in the arterial blood, osmolality of the body fluids, carbon dioxide concentration, and perhaps various other functions. These chemoreceptors include receptors of the taste bud, olfactory epithelium, aortic, and the carotid bodies. Next, we look into the differential sensitivity of the receptors. The number of receptors are shown on the right hand side, which include the free nerve endings, Pacinian corpuscles, Meissner's corpuscles, the Golgi tendon apparatus, and various others. The first question that arises upon seeing all these receptors is that how do two types of sensory receptors detect different types of sensory stimuli? The answer is by differential sensitivities. That is, each type of receptors is highly sensitive to one type of stimulus for which it is designed, and yet it is almost non-responsive to other types of sensory stimulus. Thus, the rods and the cones of the eyes are highly responsive to light, but are almost completely non-responsive to normal ranges of heat, cold, and pressure on eyeballs or chemical changes in the blood. The osmoreceptors of the supraoptic nuclei in the hypothalamus detect minute changes in the osmolality of the body fluids, but have never been known to respond to sound. Finally, pain receptors in the skin are almost never stimulated by usual touch or pressure stimuli, but do become highly active the moment tactile stimuli become severe enough to damage the tissue. Now, another question comes into the picture is how, while receiving the particular stimulus from the external environment, the receptor informs the central nervous system that it has received a specific modality of the stimulus. Say, the rods and cones present in our eyes will detect the electromagnetic radiation or light coming into our eyes and send the action potential. How does the central nervous system understand that this action potential is rudimentarily different from an action potential generated by neurons present in our ear, which respond to the auditory signal or sound. This is a concept which is known as the labeled line principle. Each of the principal types of sensation that we experience, pain, touch, sight, sound, and so forth, is called a modality of sensation. Yet, despite the fact that we experience these different modalities of sensation, nerve fibers transmit only impulses. Therefore, the question is, how is a different nerve fiber transmit different modalities of sensation? The answer is that each nerve tract terminates at a specific point in the central nervous system, and the type of sensation felt when a nerve fiber is stimulated is determined by the point in the nervous system to which the fiber leads. This specificity of nerve fibers for transmitting only one modality of sensation is called the labeled line principle. Thus, essentially, the signal itself is not important, right? The location where the signal is being transmitted in the central nervous system is important. This was very elegantly described by certain experiments in which, during brain surgery, when certain parts of the brain was stimulated by external sources, the sensory receptors which terminated in that part of the brain, the person whose brain was being excited could feel the sensory receptors that terminated in that part of the brain to be excited. So it can be imagined as the person, if a touch receptor area or the somatosensory cortex of the brain was being excited externally by direct stimulation of the brain, the patient will feel as if he's sensing touch on various parts of his body without actually the receptors responding, but essentially the central nervous system portion that to which the nervous signals are transmitted is the one that is being is the one that is responding directly. Next, we look into transduction of sensory stimuli into nerve impulses. Local electrical currents are at nerve endings, which is known as receptor potentials. All sensory receptors have one feature in common. Whatever the type of stimulus that excites the receptor, its immediate effect is to change the membrane electrical potential of the receptor. This change in potential is called the receptor potential. We look into the mechanisms of what exactly the receptor potentials, what affect the receptor potentials. Different receptors can be excited in one of several ways to cause receptor potentials. The first one is by mechanical deformation of the receptors, which stretches the receptor membrane and opens ion channels. By application of a chemical to the membrane, which also opens ion channels. By change of temperature of the membrane, which alters the permeability of the membrane. Or by effects of electromagnetic radiation, such as light on the retinal visual receptors, which either directly or indirectly changes the receptor membrane characteristics and allows ions to flow through the membrane channels. It will be recognized that these four means of exciting receptors correspond to in general with different types of known sensory receptors. In all instances, the basic cause of change in membrane potential is a change in membrane permeability of the receptor, which allows ions to diffuse more or less readily through the membrane and thereby to change the transmembrane potential. Next, we look into the maximum receptor potential amplitude. The maximum amplitude of most sensory receptor potentials is about 100 millivolts. But this level occurs only at an extremely high intensity of sensory stimulus. This is about the same maximum voltage recorded in action potentials and is also the change in voltage when membrane becomes maximally permeable to sodium ions. When the receptor potential rises above the threshold for eliciting action potential in the nerve fiber attached to the receptor, then the action potential occurs as shown on the picture here. Note also that the more the receptor potential rises above a threshold level, the greater the action potential frequency becomes. That is shown by the peaks that are shown here. So in the first part of the plot, we can see that the difference between successive peaks or successive action potential is large, but this reduces to a very minute amount, that is frequency of action potential increases. Next, we look into an example of receptor potential of Pacinian corpuscles, which is an example of receptor function. Let us first look into the anatomical structure of Pacinian corpuscles, which is shown in the figure. Note that the corpuscle has a central nerve fiber extending through its core, which is shown right here. Surrounding this is multiple concentric capsule layers so that the compression anywhere on the outside of the corpuscle will elongate, indent, or otherwise deform the central fiber. Now let us look in the next figure, which shows only the central fiber of the Pacinian corpuscle, which is right here. After all the capsule layers, but one has been removed. The tip of the central fiber inside the capsule is unmyelinated, but the fiber does become myelinated. That is, there is a blue sheet, the myelin sheet that covers the particular nerve fiber shortly before the corpuscle, or shortly before the nerve fiber leaves the corpuscle and enters the peripheral sensory nerve. The figure shows the mechanism by which a receptor potential is produced in the Pacinian corpuscle. Observe the small area of the terminal fiber that has been deformed by the compression of the corpuscle. And note that ion channels have opened in the membrane, allowing positively charged sodium ions to diffuse to the interior of the fiber. This creates increased positivity inside the fiber, which is the receptor potential. The receptor potential in turn induces a local current in the local circuit of the current flow, which is shown by red arrows, that spreads along the nerve fiber. At the first node of granule, which itself lies inside the capsule of the Pacinian corpuscle, the local current flow depolarizes the fiber membrane at this node, which then sets off typical action potential that is transmitted along the nerve fiber towards the central nervous system. This is how the Pacinian corpuscles detect the sensation of touch. Next, we look into relation between stimulus intensity and the receptor potential. The figure here shows the changing amplitude of the receptor potential caused by progressively stronger mechanical compression, which is essentially increasing stimulus strength, applied experimentally to the central core of the Pacinian corpuscle. Note that the amplitude increases rapidly at first, but then progressively less rapidly at high stimulus strength. In turn, the frequency of repetitive action potential transmitted from a sensory receptor increases approximately in proportion to the increasing receptor potential. One can see that very intense stimulation of the receptor causes progressively less and less additional increase in the number of action potentials. This is an important principle that is applicable to almost all sensory receptors. It allows the receptor to be sensitive to very weak sensory experience and yet not reach a maximum firing rate until the sensory experience is extreme. This also allows the receptor to have an extreme range of response from very weak to very intense. Next, we look into adaptation of receptors. Another characteristic of all sensory receptors is that they adapt either partially or completely to any constant stimulus after a period of time. That is, when a continuous sensory stimulus is applied, the receptor responds at a high impulse rate at first and then at progressively slower rate until finally the rate of action potential decreases to very few or often none at all. Figure shows the adaptation of certain types of receptors. This figure right here. Note the Pacinian corpuscle adapts extremely rapidly and hair receptors adapt within a second or so, whereas some joint capsule or muscle spindle receptors adapt slowly. Furthermore, some sensory receptors adapt to a far greater extent than others. For example, the Pacinian corpuscle adapts to extinction within a few hundredths of a second, and the receptors of the base of the hair adapt to extinction within a second or more. The longest measured time for complete adaptation of a mechanoreceptor is 2 days, which is the adaptation time required by the carotid and the aortic baroreceptors. As we can see right in this picture, the Pacinian corpuscle adapts very rapidly, and the impulses that it transmits or the stimulus that it detects and then transmits the signal to the nervous system drops down to zero within a fraction of a second. The hair receptor also does so in a time period of seconds, whereas other receptors such as joint capsule receptor or muscle spindle adapts but continues to send signals for a longer period of time. Next, we look into mechanisms by which receptors adapt. The mechanisms of receptor adaptation is different for each type of receptor. In the case of mechanoreceptors, the receptor that has been studied in great detail is the Pacinian corpuscle that we just discussed. Adaptation occurs in two major ways. First, the Pacinian corpuscle is a viscoelastic structure, so that when a distorting force is suddenly applied to one side of the corpuscle, this force is instantly transmitted by the viscous component of the corpuscle directly to the same side of the central nerve fiber, thus eliciting a receptor potential. However, within a few hundredths of a second, the fluid within the corpuscle redistributes so that the receptor potential is no longer elicited. Thus, the receptor potential appears at the onset of compression but disappears within a fraction of a second, even though the compression continues. The second mechanism of adaptation of the Pacinian corpuscle, which is a much slower one, results from a process called accommodation, which occurs in the nerve fiber itself. That is, even if by chance the central core fibers should continue to be distorted, the tip of the nerve fiber itself gradually becomes accommodated to the stimulus. This probably results from progressive inactivation of sodium channels in the nerve fiber membrane. And thus, the Pacinian corpuscle, even though it is being stimulated, does not transmit nerve impulses to the central nervous system. Now, finally, we'll look into two separate types of receptors, which are the tonic receptors and the rate receptors. The slow adapting receptors or the tonic receptors continue to transmit impulses to the brain as long as the stimulus is present. Therefore, they keep the brain constantly apprised of the status of the body and its relation to the surroundings. For instance, impulses from the muscle spindles and Golgi tendon apparatus allow the nervous system to know the status of the muscle contraction and load on the muscle tendon at each instant. Other slowly adapting receptors include receptors of the macula of the vestibular apparatus, pain receptors, baroreceptors of the arterial tree, chemoreceptors of the carotid and the aortic bodies. Because the slowly adapting receptors can continue to transmit information for many hours, they are called tonic receptors. Next, we look into rapidly adapting receptors which detect change in stimulus strength, which are known as the rate receptors, movement receptors, or phasic receptors. Receptors that adapt rapidly cannot be used to transmit a continuous signal because these receptors are stimulated only when the stimulus strength changes. Yet they react strongly while a change is actually taking place. Therefore, these receptors are called rate receptors, movement receptors, or phasic receptors. Thus, in the case of Pacinian corpuscle, sudden pressure applied to the tissue excites this receptor for a few milliseconds, and then its excitation is over even though the pressure continues. But later, it transmits a signal again when the pressure is released. In other words, the Pacinian corpuscle is exceedingly important in apprising the nervous system of rapid tissue deformations, but it is useless for transmitting information about constant conditions in the body. That was all we had for today. These are the references to the slides we have discussed today, which are the chapters 45 and 46 from the textbook of medical physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to this video.